Well, good morning, everybody. Or I could also say good afternoon, as we have participants from different time zones joining us here today. Just a very short welcome on behalf of the Danish-Swedish Life Science Cluster Organization, Medicon Valley Alliance. My name is David Muniz-Sepanek. I am the head of business development and public affairs with Medicon Valley Alliance. I truly hope and believe that you will enjoy today's webinar with the title Customized Services of Antigen and Antibody Production, co-organized with our member company, Sino Biological, and the team consisting of Dr. Chen, whom you will meet shortly, and Sales Director Bin Hu, and a Key Account Manager Nordics, Yi U. Will you just shortly say hello and introduce yourselves?
Of Sino Biological Europe, and thank everyone for joining us, and really thank Lico and David for your excellent organizing effort. But it's just a pity that right now we can only meet here online, but we do hope in the near future we can visit at least some of your companies in person at Medicon. And today we are here basically open for any kind of questions and for the discussion of any kind of cooperation models. Okay, thanks, and back to David. Yeah.
Hi. So good morning and welcome, everyone. I'm Yi, the Key Account Manager from Sino Biological. I'm responsible for the inquiry from Nordic regions. I hope you all will enjoy this section, and please feel free to contact me in the future if I can be of any assistance. Thank you.
Thank you very much, you and Sales Director Bin Hu. And then just back to the introduction, obviously, if you get inspired not only by the content of today's webinar, but also by the concept, and start to wonder if that would make sense for your company to organize something similar with Medicon Valley Alliance, feel free to reach out to me. You will find my contact info on our webpage. And it will also appear on the final goodbye slides after the presentation and after Dr. Chen's Q&A session. If you have any questions about Medicon Valley Alliance membership, how to join Medicon Valley Alliance networks, which employees of member companies can do free of charge, don't hesitate to give me a call or send me an email. I also encourage you to go to our webpage and visit the event calendar, where you'll find a number of hopefully interesting and inspiring online and offline events organized by either Medicon Valley Alliance or some of our member companies and organizations. And I would especially highlight our upcoming annual meeting, Accelerating Growth in Medicon Valley, Bridging Science, Innovation and Healthcare, which takes place at the Clarion Hotel, Copenhagen Airport, on the 8th of November, and consider yourselves invited. You can register free of charge on our webpage, and you will find the event listed in the event calendar. A few practical remarks for this webinar. You can say our household rules for today. It's pretty much standard operating procedure, but as you can see, you are automatically muted. You should use the chat for technical questions only, and then we use the Q&A for questions to our speakers, that is Dr. Chen, and then be aware that the event will be recorded, and you will actually receive a follow-up email with a link to the recording and an opportunity to provide us with some feedback about the webinar. Yes, enough said on my part. I am not the reason why you tuned in today. So fasten your seatbelts and let's get started. And then without further ado, over to Dr. Chen, who is actually the reason why you tuned in today. Dr. Chen, the floor, or in this case, the screen, is yours.
Okay, thank you, David. Thank you for the introduction, and good morning, everyone. And thank you for joining us today for this webinar. My name is Yuning Chen. I'm currently the Research and Development Manager here at Sino Biological. And I would like to take this opportunity to thank Medicon for organizing this amazing event. And I hope you will find something interesting regarding what we do, and hopefully to encourage discussion as well as future collaborations. Okay, so a quick overview of our company. Sino Biological was founded in 2007. Our headquarters is located in Beijing, China, and that's also where all the manufacturing facilities are. And we also have branches in Shanghai and Guangzhou, which covers pretty much all the mainland China territory. And we also have offices in somewhere in the United States, and I think the European office is located in Frankfurt to handle global business. So our company is actually growing pretty fast, and we are recently rewarded an IPO in kind of equivalent of the United States NASDAQ. So we have now more than 450 employees, and 30% of our staff has advanced degrees, either master's degree or PhD. We have a large lab office space as well as GMP facility located in Beijing, as I mentioned earlier. Our core business, including recombinant proteins, antibodies, and gene clones and ELISA kits as reagents, and we also provide customized services to generate these biologics, essentially, and this is going to be our main topic of the day. So before we get into the service, I do have to mention the products that we carry. We carry over 28,000 genes and over 6,000 now, I believe, is over maybe 7,000 proteins, and also antibodies of various kinds, as well as ELISA kits. Our products are very well received, especially, and they're generating many citations during the past 10 years. And our customers, our end users are distributed in more than 90 countries worldwide. So we've been doing this for over a decade. So we do have quite some experience in recombinant protein and antibody expression and manufacturing. So in terms of CRO services, which is our main entree of the day, if you will. So this is a slide that just briefly introduces what we can do for customers overseas at this point. So basically, we utilize our recombinant protein expression service platform. We offer recombinant protein expression service as well as recombinant antibody production service. And with our antibody development platforms, we will also offer antibody discovery services as well, which I will elaborate later. So I think I really don't want to make this webinar as a pure sales pitch. So I think instead of, along with the brief the general introductions, I will also include a few cases, showcasing the work that we did in the CRO service area to give you guys a more comprehensive understanding of our capabilities. Okay, now let's move on to our first main topic of the day, which is recombinant protein expression. So recombinant proteins are very useful tools, not only in the realm of scientific research, but I think therapeutics derived from recombinant proteins or recombinant antibodies, which are essentially recombinant proteins of their own rights, are becoming a more and more important part in people's lives and in our battle against various different diseases. And comparing to natural proteins, recombinant proteins have, of course, they have significant advantages, including unlimited resources, and they can be made as animal-free manner and have higher yield comparing to their natural counterparts, and their batch variants can be very well controlled thanks to the development of modern bioreactors and controllable bioreaction vessels. So basically, in order to generate a recombinant protein, we take a target gene, clone it into a expression vector, and then we transfect the expression vector into various host cells, and then the vectors will harness the cellular machinery of the host cells to generate the protein. And once the proteins are generated, they can be purified using chromatography approaches to get the purified protein. So here at Sino, we have 4 major platforms for recombinant protein expression, which including the mammalian systems such as HEC293 and CHO. The proteins expressed by these platforms are usually have correct or close to natural post-translational modifications. Well, given that they're eukaryote proteins. And they're very good at producing soluble proteins, but one caveat, maybe if you will, is they require long culture duration and is relatively more costly than platforms such as E. coli. And insect cells are also useful tools for recombinant protein expression. They can be cultured to a higher cell density, so that they're very good for expressing intracellular proteins and membrane proteins as well. But insects have a unique profile of glycosylation profile, so the proteins they produce often have shorter glycan chains. But this could also serve to some advantage if you're working in the field of protein crystallography and to try to obtain a crystal structure of a protein. And sometimes heavy glycosylations could be problematic in order for the proteins to pack and form crystals. And then of course there's E. coli, which is a prokaryote system. It can produce proteins at relatively rapid scale and low cost, but these proteins produced by E. coli usually doesn't have any post-translational modifications. And sometimes when the proteins are massively made, they can go into inclusion bodies, which will add some difficulty in protein expression. So the host cell selection is actually very important to I would consider it as the first step in order to obtain a successful recombinant protein. And once we received an inquiry, we will perform a sequence analysis of the protein and try to identify these parameters. There are the parameters of interest, and which will dictate the protein construct as well as the downstream processes associated with the expression and purification of the protein. The basic parameters such as molecular weight and isoelectric points will determine what kind of a purification method that we're going to use, and some structure features on the proteins. For instance, if what kind of post-translational modifications that we might have, and sometimes if the protein have some motifs such as GPI anchors or maybe transmembrane domain, we would consider removing these parts in order to make the protein expression a little easier. And cysteine content profile is of course is another important aspect of the protein analysis. It will allow us to determine how complex the higher order structure of the protein is, and then to select the most suitable host system to express these proteins. And the other parameters such as instability, hydrophobicity distribution, and disorderness will also dictate if this protein is going to be easy or hard to express. And and we will review all this information and come up with a proposal, which will include what kind of expression host that we're going to choose, what construct, which means which section of the proteins or the full length of the proteins that we're going to make, what kind of affinity tags that might be associated with the protein to facilitate purification, and also, of course, what kind of quality control methods and what's the quality control standards that we're going to pursue and during the process of protein expression. And after that, we're going to run a pilot we're going to run a pilot expression to assess the feasibility to see if the protein is feasible. If everything works well, we can scale up the expression and the delivery of the proteins. Here is a pie chart showing the host species of proteins of interest that we made during last year, 2019 and 2020. As you can see, the human proteins, actually, of course, they're the major market share, but since fortunately or unfortunately rather, viruses were the star, quote unquote, star of last year. We had a lot of requirements for virus proteins as well, especially the ones of COVID and influenza, and we also express proteins from other species as well. So we do. This is highly customer oriented, and we do have some experience in proteins derived from different species, and these and we also have extensive experience in producing proteins from upper respiratory virus strains, and these proteins have been formulated into this high throughput chip-based assay platform. Which, if you're interested, you can look up this Sinomium, Sinomium chip, and hope it will be helpful to you as well. So affinity tags, they're important in order for both protein purification as well as sometimes for enhancing solubilities. So in here lists all the most of the common protein fusion tags that we use. At Sino, we mainly use his tag and the FC tag as purification tags, but other protein tags are also available to facilitate the solubility or purification of the proteins as well. Please do pay attention that if you would like to maybe fuse your protein with a protein tag, for instance, GST or MVP, or even the FC tag, they could sometimes be problematic because of their size, and they could impose some space hindrance, and sometimes might even mask the active site of your protein. So if a soluble tag or a protein tag is necessary, you might want to consider at a protease cleavage site between your target protein and the fusion protein in order to remove the protein tag afterwards. Okay, so I would want to also share with you some cases in our during our work. A successful recombinant proteins can be made using different approaches combined essentially, and in this case it actually shows the generation of a tag-free dimeric protein using the HEC293 expression system. So this showcases the purification steps that we undertook in order to obtain a protein with the 99% dimer content, as well as to retain its biological activity. So the first the first purification, we used a ion exchange, and then we enriched the fractions containing the protein of interest using 2 steps of gel filtration columns, which resulted in the protein with 99% dimer formation. And also the protein is active in terms of its proliferation activity. So this means I think this case indicates that a affinity tag is not necessarily required to get high purity proteins, but that a word of caution is if the protein is expressed at a low level, then to obtain a tag-free protein might be challenging because it will require multiple steps of purification, and during each step there might be some losses associated with the purification. Okay, so the second case I would like to show how important the correct construct is going to impact protein expression. So this one is the extracellular domain of a single pass membrane protein that we made in insect. So in the first construct, we tried to secrete this protein into this culture supernatant, but observed there was no secretion at all during the first construct. Then we went back and performed a sequence hydrophobicity analysis and identified there is a highly hydrophobic region at the N-terminus of the protein, which could be problematic and causing it not to be able to express or to form aggregates and then degraded by the host cell. So after a discussion with the client, we agreed that this section is not relevant to their desired functionality. So we went ahead and removed this section. And the revised construct, as you can see, showed a clear protein secretion, and we obtained a very good monomeric protein after just one step of nickel affinity purification. So so the constructs are important, and certain regions of the protein might need to be removed in order for it to be expressed. And here also the third case is actually we expressed a single domain membrane protein, and during the first extraction route, we used a certain combination of buffer and detergent, which failed to extract the protein. And then and then we went back and looked at alternative approaches in literature, and then came up with the second combination of detergent combinations. And you can see that this combination did successfully extracted the protein in this elution 2. And after a polishing step, after a gel filtration polishing step, we have a very good single pass transmembrane domain purified and stored in a buffer that contains both PBS and DDM, which is another detergent to stabilize the protein. So I think in this case, shows that detergent selection is very important for the successful extraction of membrane proteins. Okay. All right. Last but not least, this case is actually something that we take on during our product development. So we're trying to express the nucleocapsid protein from SARS-CoV-2. Initially we succeeded in producing this protein using insect cell system, but due to some reasons, the proteins are existed in a mixture of monomers, oligomers, as well as aggregates. So we switched the expression system to E. coli, and during the trial with the first strain, we do see the expression of the protein, but the protein is degraded easily, which indicates there are some structural defects in the proteins produced this way. And then we moved on to the second strain. Since E. coli, there are a lot of engineered strains available. So this is also a good optimization strategy, if you will, in terms of recombinant protein expression. And we switched on to the second strain, and you can see the degradation phenomenon is resolved, and we obtained high purity protein at a correct oligomization status as assayed by gel filtration chromatography, and the protein is actually very stable and can survive 3 times or more freeze and thaw experiment. So this case indicates that the expression host is also a very important plays a very important role in obtaining correct recombinant proteins. All right, so. And then now came to the second part, which is a recombinant antibody expression service. So antibodies are becoming more and more important in literally all aspects of life science, and they come in different subtypes and subclasses. So we have experience with all subclasses of IgG, and we also have experience in the production of IgM as well. And I think also in the horizon are these fragmented antibodies, which include Fab, SCFB, and single domain antibodies, as well as antibody fusion proteins, and these I call them combinational antibody, which either a chimeric antibody, which has the various region of one species grafted onto the constant region of another species, or bispecific antibodies that comes in even more shapes and formats. So we have experience in actually expressing all type of these fragmented antibody fusion proteins, as well as the chimeric and bispecific antibody as well. So here's a slide that generally summarizes our general approach and also showcases some data from our previous projects. So basically, for the expression of a conventional antibody, we clone the antibody into two vectors, one harboring the light chain and the other harboring the heavy chain. And we co-transfect these vectors into a host cell, normally HEC293 or CHO, and the cells will express the antibodies, and we can purify antibodies using mainly protein A or protein G affinity chromatography approaches. And you can see we can make proteins at high purity and good monomeric dispersion, despite their subclasses, either a mouse IgG-A or mouse IgG1 or mouse IgG2A or 2B. And here on the right corner, it shows the successful cases of recombinant antibody fragments, either fused with a his tag or fused with an FC tag. And up on the right corner summarizes the cases of different formats of bispecific antibodies that we produced during the course of, I believe, last year alone, or last year alone. So you can see we have some certain project experience in these formats, but we also welcome to welcome other formats to I think together with our client to enhance our capability, and also in the meantime to try to deliver you the product that you desire. And one of the featured services that we provide is to use a FUT8 double knocked out HEC293 or CHO cell lines to produce afucosylated antibodies, which means the fucose residue on the antibody glycan chain will be removed using these cell lines. And these afucosylated antibodies are very good for ADCC activity enhancement. And here showcases two antibodies expressed either in the FUT8 double knocked out HEC293 cells or the double knocked out CHO cells. Basically, you can't really see a difference in molecular weight or monomeric dispersion of the antibodies by SEC analysis alone. But when these antibodies are examined by mass spec, we clearly identified a molecular weight shift that corresponds to the absence of the two fucose residues on the glycan chain. So, if the ADCC performance is something that you're interested in, these cell lines are very good tools to generate ADCC enhanced antibodies. And another feature service for recombinant protein expression is to produce IgMs, which are either pentamers or hexamers, so they're giant molecules. But we have established two methods to produce pentameric IgM. So basically, I think the success of obtaining the pentameric IgM is to incorporate the J chain into the expression system. So the two methods listed here, we have different cloning strategies to introduce the J chain into the mixture. And you can see we obtain high purity IgMs with correct oligomeric status as assayed by SAC chromatography. For the hexamer, the J chain is not required. So just a co-expression of the light chain and the heavy chain is adequate to generate the high molecular weight hexameric IgMs at high purity. Right. So I think as for in the realm of services, the lead time is also very important for you. So here I actually list the the lead times that normally take for each platform to express proteins proteins and antibody in the case of the mammalian culture system. So please do note that if you have a large gene, then it might require a little bit longer for the gene synthesize. But once the gene is made, the rest should all work as planned. So normally it takes about 6 to 8 weeks to generate a recombinant protein, and for insects it may take a little bit longer since it will require a virus packing process. Okay. So another featured recombinant protein or antibody expression service that we offer is this high throughput expression platform that we established a while ago. So basically what happens is we take the antibody or we take the protein sequence library, then we design the primers for this library, and we can make vectors in this high throughput format. Then we transfer these vectors into the HEC293 culture array. These cultures can be from a volume of 20 mL to 400 mL in the shaker flask. And then we can perform like one-step purification to obtain protein molecules or antibodies in this library, and then we can send them for client's validation. And for the ones that you decide to proceed, we can just directly transfer them into the bioreactor and for larger scale manufacturing. So far the data is pretty good for this platform. We can produce 100 to 200 antibodies or proteins per week, and the average antibody production success rate is at about 99%. So we optimize the process, so it will require only 2 to 4 weeks from gene synthesize to purified protein or antibody. So we've also used this platform to produce virus proteins, for instance, the HA or NA from influenza, and the RBD mutations from SARS-CoV-2. And this platform also actually revealed very robust results for producing these proteins as well. So here's an example. So we contracted a project from a client, I think at the beginning of last year, which is in the beginning of the pandemic. So they actually extracted B cells from COVID-19 patients and generated antibody sequence libraries. And we created about 600 antibodies for them, among which they performed high throughput screening and identified 14 neutralizing antibodies. And then we crack up the scale and produce a large quantity of these antibodies for them to perform either structure analysis or animal studies. And the results are published in Cell, I think, in May last year. So the lead time for high throughput antibody expression is a little bit over 3 weeks, and we can express antibodies using in the IgG1, 4, and IgG2 subclasses. And standard quality control usually involves UV for quantification as well as purity determination by SDS page. But other assays can be included as well, including the SEC HPLC endotoxin analysis, or some binding or affinity assessment by other high throughput methods such as BLR or SPR as well. So please note, if you would add some optional analysis, it would prolong the lead time of such projects. So so I think before are the services that we offer using the recombinant expression technique, essentially. And I think during the course of our company, we also established robust antibody development platforms, and these platforms have given us over, I think, 10,000 different antibodies as our catalog product. And I think, and now these platforms are also open to contract services to generate customized antibodies for a customer's specific purposes. So I think we have a very comprehensive animal immunization regimen, immunization platform, antibody generation and screening platform, as well as these assay platforms to identify the most suitable leads for us. And our experience ranged not only from classic mouse monoclonal antibody development using hybridoma, but also we have established a phage display monoclonal antibody development platform, as well as a single B cell based antibody development platform as well. So, and we also have experience developing antibodies against a specific post-translational modifications. So, for instance, phosphorylation. And we also have experience in generating anti-idiotypic antibodies for, I think, purposes such as ADA assessment and so on. So, as I mentioned earlier, we can use different format of antigen to generate antibodies. So during our course of both product development as well as service, contracted research service, we have used antigens in the format of peptide, and we routinely using recombinant protein as immunogen, but we also have experience using DNA encapsulated in the lipid vesicle as antigen, as well as using the whole cell. But the DNA and whole cell immunization is a little bit trickier, since especially for the whole cell, since the cell will create a significant amount of quote unquote background. So in order to screen and identify suitable candidate targets, it will require either a negative cultured cell line or cultured cell lines that does not have the protein of interest, or it will have we will also require a certain format of recombinant format of the target protein in order to identify the most suitable leads. Okay, so yeah, so this is just some I think some tips that we learned from our past experiences. And we have these antibody generation routes as listed here: hybridoma, phage display, as well as B cell sorting. And we also have, of course, we can also provide polyclonal antibodies. And the antibodies that we generate are, as you can see from our catalog products, they're excellent for applications such as Western blot, ELISA, or we can make ELISA pairs, as well as they can be used for immunofluorescence, immunohistochemistry. We have quite some experience with that regard as well. And also these antibodies, some of the antibodies are proven to be able to use in cellular function assays and FACS, as well as to serve as affinity purification reagents. So a variety of antibodies can be generated using our platform. So generally, it will take a little bit longer, of course, to generate antibodies from scratch. Essentially, I think the immunogen preparation will cost some time to do so, and the animals will require some time to mount a sufficient immune response, and after which they can be sacrificed and to proceed into the antibody selection process. But generally, it takes 4 to 6 months maybe to complete a mouse or rabbit monoclonal antibody generation service. But we do have a speedy immunization regimen, which combines our unique immunization strategy as well as adjuvants, so that we can expedite this process and finish a project in about 3 to 4 months time. So this is also an option here. I do understand that time is sensitive in any I think research and development efforts. So we're also trying to work as quickly as possible in this regard. So here showcases a recent case that we performed for a client to develop an antibody using a single B cell sorting technique. So basically, we have identified, and after the sorting of the B cell, we identified 380 antigen-specific B cells, and we then generated about 42 sequence pairs, which yield about 46 clones, and among which 8 are better than the control antibodies that are commercially available. So I think after immunization to all the way to antibody characterization, it took us about 26 days. So the B cell sorting platform is without doubt an expedited method to generate customer antibodies. So last but not least, I would like also to give a few minutes to introduce our CRO service for virology research. So as I mentioned earlier, unfortunately, due to the SARS-CoV-2, I think the world is more and more aware of the threat of viruses, and in order to fast respond, current pandemic or maybe future pandemics, hopefully not. So tools are critical in this regard. So we have developed a ProBio virus research reagent banks that covers that contains a lot of reagents with and that covers about over 50 virus currently known virus strains, and we can generate recombinant antivirus proteins at very fast fashion. We generated influenza proteins in about 12 days, and the SARS-CoV, it only took us 9 days to generate the SARS-CoV-2 and I think the alpha variants RBD version of the SARS-CoV-2 protein. So so the faster response, and we also have a comprehensive collection of virus antigens, and here you see again this Sino chip, which contains many upper respiratory virus antigens. And we also have a collection of influenza vaccine strains from 2015 all the way to 2021. And also the SARS-CoV-2 spike mutants are one of our featured library as well. We have now I think we have over 180 RBD or spike protein mutants in this collection, and we also have antibodies with hypersensitivity against these virus proteins as well. So I think due to the limitation of time, I don't want to go into too details on this slide, but I think it shows the take home is we have a we have almost completely mapped the proteome of SARS-CoV-2, and with focus on the spike protein, and have generated over 170 RBD mutants. They can be used to for maybe antibody screening to identify antibody evasions. And here are two examples showing the activity of the RBD and S1 by either the Delta variant and the Kappa variant. Both proteins showed a slight increase in affinity towards the ACE2 receptor. We believe that might be one of the reasons why these Kappa and Delta variants are more potent than the quote-unquote wild type. And here we also have a full-length extracellular domain of the SARS-CoV-2 in the format of the HexaPro. I think it's a technology developed by UT Austin, and by which we are an authorized commercial partner with them. So we can make mutations, or we can make variants based on this HexaPro backbone. And so far we have made I think more than 6 mutants variant proteins. Here I only show the quote-unquote the wild type, which bears the D614G mutation, as well as the variants from Delta and Alpha. And all proteins are in the correct trimeric formation, and they're active in terms of their ACE2 binding activity. So last but not least, we do have a platform to generate high quality antibodies against SARS-CoV-2 and other viruses for that matter. So our antibodies can be highly specific against the SARS-CoV-2, but not react to other coronaviruses. They can be highly sensitive with lower sensitivity at 42.5 picogram per ml. This is anti-nuclear capsid antibody. And we can also generate neutralizing antibodies, and these can be used as controls to assess, antibody assess the antibody evasion phenomenon of the mutants. And last but not least, we also have a collection of those quote-unquote broad spectrum anti-nuclear capsid antibodies, and they can recognize different nuclear capsid protein from different variants at equivocal specificity and affinity. So these antibodies are very useful in terms of to formulating them into assay kits to identify nuclear capsid proteins from SARS-CoV-2. Okay, so I think this I think this is the one slide before the last one. So this is basically a general workflow of the CRO service. So basically, for once the inquiry is received, I think either by Dr. Hu or by Yi, they would, based on what kind of inquiry it is, it could be a standard service package. You will receive a standard quote, and once the payment is confirmed, then we can carry out the project, and you will receive updates from time to time until the completion and shipment of the project. If the project is more complicated, for instance, if the protein or antibodies generated are not very conventional, and if you're trying to contract us with for instance, a antibody generation project. So these projects are considered to be considered to be more complicated and requires more steps. So there will be more back and forth. But after we finalized with the client about the specifics, and once the payment is secured, we'll also again go through to here to start the project, and you will receive updates. So we will try to keep you informed every step of the way by our very professional project management team, and until the end. Okay, so I think that's all I have for today. Here is the contact information of our European office. So yeah, again, thank you for your time. Thank you for listening, and I'd be happy to take any questions that you might have. Okay, thank you. Okay.
Yes, and I think we do have some questions from some of our participants. We have one question here for you, Dr. Chen.
Thank you.
What is your capacity in terms of recombinant protein or antibody production?
All right, thanks, David. I was actually trying to look for where the text box is. Okay, so yeah, in terms of a capacity, I think we can provide recombinant protein or recombinant antibodies in the quantity of from micrograms all the way to grams. Depending on how much antibody or protein the client need. This is our scale. So we have small scale expression vessels such as shaker flask or small volume bioreactors, but we also have large bioreactors all the way to, I think, 1,000 liters. So that can be used to handle different requirements from clients.
Thank you. I think we have time for a few more questions. I have a second question here. You have an extensive collection of recombinant virus proteins, but how do you choose the most suitable expression host for such proteins?
Yeah, so this is a very good question. So virus proteins are kind of unique because from a revolutionary point of view, virus is not close to either a prokaryote or a eukaryote species. But these proteins are also extremely complicated. So when you look at the spike proteins of SARS-CoV-2, it's a trimeric format and with heavy glycosylations, and we do see similar complexities in proteins from other viruses as well. For instance, the glycoprotein from HIV or the HA from influenza. So for these proteins, due to their complexity and due to their highly complicated higher order structure, we would want to use a eukaryote system to produce for their production. So for one thing is to ensure the correct, ensure their structure is correct, and also to add on as much post-translational modifications in the form of glycosylation as possible. But for other virus proteins, so for instance, these for some enzymes of the virus, we find that a eukaryote expression platform such as a prokaryote expression platform such as E. coli should be able to produce adequate proteins at adequate amount and sufficient activity, for this regard. But like I showed in one of the slides, we did try to produce a nucleic capsid protein from SARS-CoV-2 using the insect cells, and we can produce the protein, but we encounter some problems in terms of its aggregation. And then when we switched to E. coli, we resolved the problem successfully. So unfortunately, there's still a trial and error process, and there's no one-size-fits-all approach. But there's some, I think, for some general guidelines, it will be. I think for the surface glycoprotein, especially for the surface glycoproteins, eukaryote systems such as HEC293 or insects should be considered first. And for the proteins involved in intracellularly or involved in the enzymatic activity side of the virus biology, then E. coli should be adequate for their production.
Okay, thank you very much, Dr. Chen. And I actually think you have succeeded in making some of the participants here curious about antigen and antibody production and your company's expertise, because the third question here is, in what format are the recombinant protein and antibody reagents supplied?
They can be supplied in basically in a liquid form. Well, I wouldn't say liquid format, but in a frozen liquid format, or it can be supplied as a lyophilized powder. I think for logistic issues, for lyophilized powders are much more easy to transfer and much more robust. However, unfortunately, not all the proteins or antibodies can survive the lyophilization process, because it's basically a dehydration process, and sometimes you will compromise the structure of the protein, causing it to aggregate after you resolubilize it with a buffer. But there are some additives that can be used to serve as protective agents, but what we do is we do a small scale trial run first to see if the protein is stable or is safe to be processed in the lyophilization manner. If it is, then we can go ahead and make it into a lyophilized powder. But if it's not, then we can ship the protein in its most stable in a buffer that retains its stability. So. But I think logistically, lyophilized powders is the easy way out. But we do try to deliver a client with the best quality protein possible. So.
Thank you. A fourth question here: is it feasible to transfer hybridoma cells to Europe?
Yeah, though I think this is also a logistic issue. Since I think every country, so I'm not very familiar with the logistics of Europe, but I think we can export biologics such as recombinant proteins, recombinant antibodies, and cell cultures essentially to Europe. I think that's logistically feasible, but it will just add some significant amount maybe to the shipping bill, and also, but however, I do have an impression that some form of biologics, for instance, tissue cultures, or some format of bodily fluids, which might contain some pathogens. These we will not be able to export to Europe. But the other way around, I think we do have projects, especially clients in China. They have generated hybridoma cells, and they want us to produce antibodies using these hybridoma cells, because we have somehow a culture system that is compatible with mouse hybridoma. So for domestic projects, there's no problem for the logistics. However, I do know that because of the restrictions of the Chinese customs, any cell-based substances cannot be imported to China. So I think I would encourage you to keep that in mind. But alternatively, I think what we could do is we could sequence the antibody from the hybridoma, which is a technique that is very well established nowadays. And with the sequence, we can generate a recombinant form of antibodies. So that will, circumvent the logistic issue. And in the meantime, you will have, the client will essentially have an unlimited supply of antibodies, and they don't have to really worry about, if they, lose their hybridomal cultures or not. And sometimes, hybridomas can secrete antibody for some time, but even though they're immortal, some hybridomas do stop secreting antibodies after certain passages. So to obtain a sequence of antibody, I think is also a very good approach to overcome this hurdle. So that the client will not lose any precious antibodies they generated from their hybridoma project as well.
Thank you very much, Dr. Chen. We are unfortunately running out of time here. Thank you very much for your willingness to take questions from the participants. And then on behalf of Medicon Valley Alliance, it's just for me to say thank you, and I hope it's been time well spent, and that you have appetite for more Medicon Valley Alliance events, perhaps even more co-organized events with Medicon Valley Alliance and Sino Biological. As mentioned earlier, feel free to reach out to me if you have any questions regarding Medicon Valley Alliance activities, our event concept, membership. And of course, I would also like to thank the rest of the Sino Biological team, Sales Director Bin Hu and Yi U, the Key Account Manager Nordics, who actually helped put this webinar together. And I guess with that said,
thank you very much.
Thank you. With that said, we have reached our destination, so to speak, and I will conclude this inspirational webinar and just say, take care out there.
Okay, thanks a lot. Yeah.
Bye, everyone.